without further ado, we'll start with 4 scientific presentation from our nodes. So each presentation is 20 minutes with questions. So if you speak less, you will have time for discussion. If you don't, we'll switch to the other one because we can't be late. So the first talk is from Anaïs Badoual from the local node, although not from the same town, from INRIA Rennes, from the Serpico project team. And I think you have the title there. And the floor is yours.
So, hello everyone. Can you hear me? Okay. So thanks for the introduction and for the invitation. In this talk, I'm going to present a framework to generate synthetic data that simulate astrocytic activity. And the goal is to support the development of a learning-based analysis tool. So here I show you an image. It's a 3D plus-time multi-slat-sheet microscopy image of astrocytes. So what are astrocytes? Astrocytes are glial cells in the central nervous system that recently emerged as key partners of neurons in information processing. So, okay, you can see the mass, but you have the part with the soma here, with the nucleus, and you can get some ramifications. And all the flashes that you see are actually the calcium signals. So they correspond to sudden and brief increases of the calcium concentration in the cytosol of the astrocytes. This is actually the way astrocytes respond to synaptic activity. As you can see in this image, there is a high diversity, a high spatio-temporal diversity of those signals. They have different amplitude, different durations, and also different frequency. And also they can be either localized within microdomains, and they also can propagate through the astrocytes. Unfortunately, today we still poorly understand the mechanisms behind those signals. We don't know if this diversity is related to different neurobiological function in the brain, and also their involvement in some disease such as Alzheimer or Parkinson. So this is why today there is a growing interest for the neuroscience community to decode this calcium code. So to do this, to decode this calcium code, we first need to extract and quantify those calcium signals from microscopy images. And as you can guess, this is a complex task, which requires the development of image analysis tools. The only thing is that today only few methods were developed and they are limited to 2D plus time microscopy images. And the problem is that when we omit the third dimension, we lose a lot of essential information, such as the number of signals occurring in a single astrocyte or where they originated. So today we crucially lack of these dedicated algorithms, and especially for 3D plus time microscopy images. And the main reason, besides the complexity of the task, is because of the difficulty to obtain large and reliable annotations that are necessary to first evaluate those algorithms, but also to train the ones that are based on machine learning. And maybe just for the story, when I joined the team, I was developing one of those algorithms for 3D plus time images. And I had to quickly stop because without this annotation, I had no way to assess the accuracy of my algorithms and to train it. So usually what we do in this context is to use synthetic data. The problem is that for this particular application, there is no synthetic data. So this is why to respond to this need, we develop a framework to generate realistic microscopy images in which we simulate astrocytic calcium activity. And to be as realistic as possible, we use the kinetic model. So in the first part of this presentation, I will show you how we simulate a single calcium event in an event. So to do this, we took a kinetic model that describes calcium events at the nanoscale that we mathematically reformulated to the macroscale, which is actually the scale that we see in images. For the kinetic model, we used a simplified version of the one that was developed by Audrey Denisot. So first, we consider that we have a stimulus, for example, coming from the synapses, at time t0 and location xc of an astrocyte that triggers a gamma increases of the calcium concentration in the cytosol. In our image, we model this stimulus by a Gaussian function, which is centered in xc and of amplitude gamma. And we limit this Gaussian function only to the pixels that belongs to the astrocytes and not to the background of the image. Then to simulate the calcium signal in response to this stimulus, we consider the following model. So first, we consider that calcium ions enter the cytosol through calcium channels that I call IP3R that are grouped in a cluster. We consider that when an IP3R is open, it releases calcium ions with a rate mu, and obviously when it's closed, they don't. The probability of an IP3R to be open has a bell shape dependence to the calcium concentration in the cytosol, which means that when you have a small increase of this concentration due to the stimulus, for instance, it will increase the probability of opening, but above a certain threshold, the probability will decrease significantly. And the last thing that we consider is that calcium ions outflow the cytosol with a decay rate of alpha. In our image, we model those calcium fluxes with this formula here on the right, where delta TE is the temporal resolution that we use to simulate our calcium event. And the last thing that we have to take into account is the fact that in the cytosol, calcium ions diffuse with Brownian motion. And we model this in our image using an anisotropic peronomallic diffusion, which is actually an isotropic diffusion, but which is maintained inside the astrocytes. And at the end, when we combine all those contributions, we simply obtain this formula here. Now, if I show you some images, just in order to visualize it. So what we do is that we created a sequence of image. We added the contour, the edges of the astrocytes, and at time t0, we model the stimulus by a Gaussian function. And by applying the formula that we just obtained, you can see the calcium signal that appears. And the last thing that we do at the end, we just remove the edge of the astrocyte to have a sequence of images with only the calcium signal. Okay. So now what I just show you is actually the simulation of one particular calcium event that is called calcium puff. So it corresponds to the signals that are really localized within macrodomain, and they correspond to the opening of one or several IP3R, but in only one cluster. But there is a second type of calcium event, which is a calcium wave. Actually, it corresponds to the propagation of a calcium puff through, from one cluster through the neighboring cluster. And to model it in our image, we developed a geometrical model that finds the center of each cluster belonging to the wave. And then at each center, we simulate a calcium puff as we just did before. Now, the final step is to create microscopy images that depict not only one calcium event, but the whole activity of the astrocytes. To do this, we use the method that I just explained to simulate different calcium events. We combine them, so this calcium event can be calcium puff or calcium waves. We combine them in a single sequence of images. for each frame, we add a background, a realistic background, which actually is a real background extracted from real microscopy images. And at the end, to recreate the experimental conditions, we added a mixture of Poisson and Gaussian noise. And at the end, we obtain our synthetic data. We developed this simulator for the ImageE platform, and we also plan to integrate it in BioImaging. And thanks to this simulator, you can generate a large diversity of synthetic data by changing few parameters that are the background, the time of acquisition, also the temporal resolution, which act as a temporal window under which calcium signals cannot be observed. And the last parameter is the number of stimuli, which control the level of activity of the astrocytes. And all the other kinetic model parameters are fixed and chosen with respect to experimental results. So here I show you some example of those synthetic data. On the left, you have the 2D plus time sequences, and on the right, the 3D plus time sequences. Here you have the real data. And the three other sequences are synthetic data, where I just changed the temporal resolution. And for the 3D, it's the same. On the bottom, you have the real data, and on the top, the real data, and on the bottom, the synthetic data. So as you can see, those images seems quite realistic, and we are under the validation phase to validate those data. I'm not going into the details of this complex validation phase, but just quickly to mention that we verified that our synthetic data, that in our synthetic data, we have all the diversity of the calcium signal. So we have the calcium puff and the calcium waves. And we also check that our synthetic calcium events are relevant in terms of amplitude and duration compared to real data. So we really hope that those synthetic data sets will be used by the community to boost the development of analysis tools, allowing to decipher the behavior and the function of the astrocytes. And just to mention that on my side, using those synthetic data, I was able to continue my algorithm. So on the top here, you can see a simulated images. And on the bottom, you have the segmented calcium signals. On the top, you also have a table with some information like the location, duration, amplitude, volume. And thanks to the synthetic data, I will be able to assess the accuracy of this information. And also, and more importantly, I was able to train a classifier to distinguish between the signals that are localized and the one that propagate. To conclude, I just want to briefly mention that these two projects, so the simulator and the analysis algorithm, are developed through an interdisciplinary collaboration between three fields: acquisition, modeling, and image analysis. For the acquisition, I work with Misa Arizono, who was at the beginning of the project in Bordeaux at the Interdisciplinary Institute of Neuroscience in the team of Valentin Nagel. And she's now in Japan at Kyoto University. And for the modeling, I work with Audrey Deniso. She is also currently in Japan at the Okinawa Institute, but she will come back in France in a few months as a junior researcher in Ria Lyon in the team of Hugues Berry. So thank you for your attention, and I would be pleased to answer some questions.
Thank you very much. Very nice presentation, and we have plenty of time for questions. So maybe I start. Actually, I have two questions. First of all, so you didn't take into account voltage-dependent membrane channels, right? There is no calcium channels with some voltage dependency in the membrane in your model.
No, actually, we really took a simplified version of the one developed by Audrey. I know that what we take into account here is that we know that in general in the cytosol, you always have some a baseline of calcium concentration. So this we took into account, but we know that you can have other facts that can modify also those calcium concentration that we for now didn't took into account. Yeah.
Okay, thank you. Before asking, yeah, one question, Lydia.
You mean to localize to know when, if the signal is from the soma, from the glial, or this is one of the goals of the project. But for now, I'm not doing this. But yeah, it will be one of the goal at the end.
Actually, my second question was actually related to yours. What determines the directionality of the propagation in your model?
You mean for the calcium wave?
Yes.
So for now, so this is the geometrical model that we designed. So we know the pixel that belongs to the astrocyte. We randomly choose one pixel for the first, for the center of the first cluster. And then we randomly choose one direction, so either going to the soma or the glial cell. And then
you choose the direction.
The first direction, if it goes to the soma or to the end of the ramification, this is random. But once this direction is chosen, it will be always going to the same direction. And with through some discussion with Audrey Denisot, we the space between two clusters and the perimeters to choose the other ones, we chose it together based on some experiments that she did.
Okay. Yeah. Thanks. Do we have other questions? Yes, Martin.
Yeah, very nice talk. How do you take into account the fact that for the segmenting, the size of the processes of the astrocyte that you see on the latest slide sheet image does not correspond to the real dimensions of the astrocyte? You have a low pass filtered segmentation from the image because you are sub resolution anyway for the diameter, the true diameter of the fine processes. So how does this go into this unknown go into the model of diffusion?
Okay. So
the question is, sorry to repeat it, many of the processes are only 20 nanometer large in the extremity, particularly these peripheral astrocyte processes. And of course, on 2-photon or lattice light sheet images, they show not at their true dimensions. So it's only what you have. But for the modeling, I mean, the result gives you right, but it's a bit conceptually tricky, I guess.
Yeah. So the thing is that this was actually the question of the background, had to have like realistic edges of astrocytes. And for this, I really use real lattice light microscopy images. So the resolution that I have on the size of the astrocytes and the ramifications are the one that I have on my images. So of course, I know that most of the calcium signals occurs in the at the end of the ramification that is not always visible even in a lattice. So this is not only a limitation of simulation, but also a limitation of the microscope.
Yeah, because it's lattice, it's not sushi lattice.
It's a lattice.
Yeah. We have, thank you. Other questions? Yeah, Lydia.
Yeah. Now, for now, I didn't simulate this, and actually, the kinetic model that I used took also the hypothesis that there are always reticulum endoplasmic in the astrocyte. And when I was talking about Audrey, she was telling me that we are not 100% sure of this hypothesis also. So I know that there are some limitations to all those models.
Okay, thank you very much. We need to stop. Congratulations again.
And so today I will present you our ongoing work trying to bridge the dimensions from the single cell, single molecule resolution up to the organoid scale. And this through a technique that we have co-developed with the team of Virgil Gasnoff at in Singapore, which is called SOSPIM for single objective selective plane illumination microscopy. Okay, so what is SOSPIM about? So it's very simple technique. So at least on the paper. So actually, it was the postdoc work of Remy when he joined the team already 10 years ago. So the idea is to use 45-degree mirrors on your cover slip in order to be able to create a light sheet which is perpendicular to your optical axis, and then using the same objective collect the fluorescent light. So this is very simple geometry, and what you see is that this little micro mirror acts as a virtual objective. So the great advantage of that is that you end with a system which is a light sheet microscope without the need of having two objectives, two or more, which is really the traditional way to create a light sheet. So what's the advantage if we now compare to traditional light sheet, which use at least two objectives? So we know the virtue of this technique for having like the very good sectioning capability, penetration depth, speed of acquisition, and also it is very gentle to live and photo bleaching. But also the fact that using two objectives also restrict, and this is known, the resolution and the use of high numerical aperture objective, and also the access and to the sample and sample manipulation, which at the end prevents to do a large amount of data. The SOSPIM, like other single objective light sheet microscopes, so they now get rid of this constraint and now put to green what was the limit of using high numerical aperture objective, and also provide access to the sample. Of course, one of the limits is the use of this micro mirror, which you definitively need to take into account. They are part of your acquisition. But you will see how we can use this microfabricated device as an advantage, more especially for keeping the focus and the registration issue. Okay, so the simplicity of the technique now is that this is really something that you can just plug on the back part of your microscope, which at the end you do not have a dedicated microscope, but you have a microscope which you add the SPIM modalities, which that you can really combine SPIM plus other techniques. So it's very simple. So you have this beam steering unit that you put on the back part of a microscope. Of course, you need this specific petri dish that contain these micromirrors. And at the end, you have a software which is role is only to trigger very precisely the position of the light sheet so that it moves along the mirror and really keep the position of your focal plane well aligned with the position of your light sheets. So it's very simple geometry. The big advantage of the technique is really the versatility, and as I said before, so it is really capable of using the same instrument to cover a wide range of biological samples from the single cell, if you use like small microwells, as you can see on the left, up to the entire organisms, if you use mirrors of about in the range of the millimeter scale. And then over the years, we have demonstrated the capability to really image from through all these dimensions, from isolated cell, live or fixed, cell aggregates, 3D cultures, which we will talk about, and then even larger specimens. And today we'll really focus on these two aspects. Okay, so now we'll start with the smaller scale, super resolution, single molecule based super resolution. So I will not go through the details. So I think you know is pretty old technique now. So just what is important is that this technique, which relies on the sparse activation and localization of fluorophore, is pretty simple to implement, but it has some constraints. And then what you need to have this technique really functional and achieve this, let's say, sub 50 nanometer resolution at least, that of course, beside the localization and to have a good labeling strategies, what is very important is that you need optical sectioning. So this is very mandatory if you want to get photons, single molecule photons above the background. And this is why this technique has been really used to image structure at a very close proximity from the cover slip, and it's very limited as soon as you want to go deeper into the sample. So it was the initial project to combine, of course, the source beam, and it's really initially why we developed this technique, to be able to do super resolution in depth using this technology. And then for this, we combine several approaches together with the source beam. So the technique we like, of course, is not restricted to the DNA paints technique, because it really offers like an infinite pool of single molecule labeling. Not prone to photo bleaching, but as you can see, that the point with DNA labeling is that if you don't have optical sectioning, you will have a huge background. So it means like you cannot go for with wide field illumination or something that otherwise you will shine all your fluorophore, which create a big background. So all the paint technically like that. So this was why we really wanted to combine here, and then to get 3D localization, we use the astigmatism. Then, of course, we have like a few problems to get rid of. So the first thing is that a design numerical aperture, which is required to collect single molecule, of course, they're more prone to aberrations. So the first thing is the first combine with adaptive optics to be able to correct for aberration and to put like a very well controlled astigmatism. And then at the end, this is what happened is that a few 10 micrometer away from the cover slip, you can see the very good sectioning, and then this nice astigmatism-based localization from which you can reconstruct the whole topology of your cell at the nanoscale level. So that works pretty well. And then on top of that, you can make sectioning. So again, it's very straightforward, at least now, but then you also have to overcome another issue, see, which is the drift. So these acquisitions are very long, more especially DNA planes, several planes. And then there is another issue with this geometry is drift. So usually axial drift is possible to correct optically, to use perfect focus system, for example. So that works pretty well, but lateral drift is something which usually is not an issue, but with the source beam is an issue. So if you just like a drift in a perpendicular direction from the mirror, then your light sheet by construction will just defocus, and then you won't have anymore the alignment of the focal plane together with the imaging plane. In this case, of course, your equation is screwed up. So then we say, okay, we have this micro mirror, we have this polymer here, which protect the mirror. So then we can put some fiduciary markers here. So by putting these fiduciary markers, now we are able, by feedback loop with a software, to be able to localize the markers which are surrounding around outside of your sample of interest, and then you are capable to feedback loop on these markers and do like an autofocus, real-time autofocus, which we demonstrated to have like a 30 nanometer axial resolution and about 15 nanometer lateral, which at the end provides even better focusing in 3 dimensions, so repositioning in 3 dimensions, even compared to the optical system. And at the end, we are now capable to have the whole entire workflow to be able to collect several planes, which can rest for hours, more especially with DNA paint, and reconstruct the whole cell in 3D, including a bit of readjustment offline, realignment offline of the sections at the nanoscale. So the thing is that another important activity in the team is the analysis. So this is the work which is really developed by Florent in the team. And now that we have this capability to collect like millions of localization in a real 3D volume, so there was a need to be able to visualize them, manipulate the data, but also analyze them. And then, so Florian, back in the days, developed this tessellation suite that we had to push for 3D. And now we have this new software, which is released and fully available, which is called POCA, so for point cloud analyst, and which has been recently accepted for publication. So the idea is that it comes with a brand new engine, which has been redesigned, very powerful, that allows manipulating millions of localizations in a very friendly manner, reconstruct, but also integrates the state-of-the-art analysis tool like the Voronoi in 3D that you can reconstruct in just a couple of minutes, even for millions of localization, from which you can extract group localization depending on different parameters. So typically here, the density, extract the clusters, manipulate the clusters, get the statistical information. So everything which was for 2D now is entered 3D, and also something which is now very interesting is look at the topology of your cluster in the 3D volumes, and then make some let's say other scale, mesoscale analysis on how your clusters of molecules are organized, typically here at the surface of the cell, and analyze there whether you have like a real polarization in the 3D, something which was not, of course, capable or needed before in 2D, but now that we have this real 3D distribution. Okay, so now, sake of time, we'll quickly jump like an order of magnitude bigger. So how can we, with the same technique, being able to investigate like this 3D culture? Like we can call like this organotypic culture, which calls spheroid or organoids. So for this, our friend in Singapore have developed this little microwell, which is called now GWell. So it's very similar to what we had before, but now is that is the specific shape where he has this four shape, is like a truncated pyramidal shape here that you can see, in which that can host your 3D sample here. It can be like a 3D culture, but also because of these four mirrors allow you also to make the source beam and the SPIM imaging. So the big advantage is that we have like all-in-one, the culturing vessel together with an imaging device. And another big advantage is that these will not to have one of these, but there are like hundreds, thousands of these microwell, which are really at close proximity one to each other. You have a matrix of this one, which make the technique now suitable for high content imaging. And here, typically, depending on the size of these microwells here, you can see that we can have like in just like 4 millimeter by 4 millimeter, the equivalent of a 96 well plate. So we have something very miniaturized, which allow to go very fast through your different 3D culture. And then from that, you can, of course, as I said before, you can combine, do some transmission lights, detect where is your organoid, and then after that, you make like this multicolor imaging, whether it is live or fixed. So they demonstrated that it's not only a system for imaging, it's also a system for culturing this 3D device, and then assess that show that there are large diversity of systems that we can culture, and just need to adapt the size of your GWell depending, and also the shape. You can have like a square, which is the most useful, but you also have an elongated one, which allow you to grow this organoid and to image them from the beginning up to a very long period of time, and then they can really grow for weeks in there, and then you can image them. So the imaging, which was the work of Tom, the team that just defended his PhD a couple of weeks ago. So the idea is now that we have this system, which is very simple in terms of image analysis. We have this array of 3D cultures that are very easy to detect just by bright field imaging. You can initiate your screening, have a little bit of interaction to select which organoid you want or you don't want. And at the end, still this issue, because now we'll navigate through them, that you need to have this light sheet perfectly aligned with the mirror. So we just, by using the transmission light image, we can use that to make like an autofocus to the system to each time reposition the light sheet at the perfect position. It's done by a very simple cross correlation, which has a resolution of, let's say, in the range of 50 nanometer, which is way enough what we have. And then altogether demonstrate that we really now have a functional high content imaging platform. And here, this is one of the first image that we did, like human embryonic stem cells. After several days of differentiation, so we could collect like hundreds of organoids in less than an hour, 3 colors, and like in a 3D volume of about 70 to 80 micrometers thick. So it's really like a big breakthrough here. And then the thing is that we have like subcellular details. System is also very good for live. So here we also proof of concept. FG2 stable cell line which express GFP, and here what you can see is that we could monitor the evolution of this 3D spheroid for several days, 20 position every 20 minutes without noticing any change, or this visualizing cells still divide, no obvious bleaching, and so that's really again just proof of concept. And finally, just relying, so I have plenty of other things I don't have time to go through, but just always now we are taking benefits of the four mirrors, which is something very usual in the light sheet. Usually, when you start to have like an organoid, you start to be pretty large. What happens is that it's pretty hard to get the whole volume if you just have like a Gaussian light sheet. And usually what you end with is that for a different reason that your sample will be sharp in one side and a bit blurry on the other side. And the strategy usually to overcome that is to use several objectives, which is of course costly, and also the alignments is a big issue. And here it's very natural that you can really combine these three things and get the best of it. So the idea is that now we're capable to get the really isotropic, let's say, on the whole side in 3D, and which we now demonstrate that is really help for the analysis behind. So with this, I'm done. Not too late, I hope. This is the people I would like to acknowledge for the funding, but also people from the team, and many other people that I didn't have time to go through, but that I hope you will hear soon, which are mostly working on all the analysis pipeline of this type of thing. Thank you very much.
Thank you, Jean-Baptiste. Yeah, we have time for a few questions. Yes. Questions for Jean-Baptiste? Oh, I actually have one question, at least one. Regarding DNA paint, that's data on fixed cells. And so I've actually always wondered, what's your experience in terms of actual access of the DNA probe in the various compartments of the cell, and how this may be hiding actually some deep compartments? Is that something you've looked into?
It's true that we're more using like simple model that we know works mostly on just for the experimental assay. What we've done, which start to work, didn't show that we could do DNA paint on like this 3D culture, for example. So it means that we can paint
fixed, permeabilized, fixed permeabilized. Yeah.
But we didn't try more than that
to know what's exact. I'm always wondering what's the diffusion properties of these DNA strands in this complex environment. Questions? Yes.
Can you give us some information about the fabrication of the micro mirror? How complicated is it, or I mean, how do you do that? Do you need a white room or this kind of things?
So yeah, the point. So so this is mostly so first we have several. That's one of the bottleneck of the techniques. So at least the production for own production is okay. For a couple of collaboration is okay. But we have no industrial way to fabricate them on the large scale. So right now it is done in the lab with pretty simple equipment. So if you have more details, maybe you can ask to Rémi. So that's really something that can be done in the lab. Once you have the main wafer, that's something we can do in the lab with no big equipment. But yeah, the large scale. Which is not
Thank you. 9 seconds left. No questions. Thank you very much, Jean-Baptiste. Yeah. And so the next speaker is Vincent Guen, I pronounce well. Vincent Guen from CRCI 2NA, Bretagne-Loire Node. Going to talk about multiscale and multidimensional imaging of the mammary gland, and that reveals the key role of the primary cilium in mammalian stem cell biology.
All right.
Okay.
So I'm Vincent Guen. I'm an interim investigator in Team 7 in the Cancer Research Center, which is located in this building. And I'm pleased to participate to this fantastic meeting. So I lead a small group that is interested in the biology of epithelial plasticity and primary cilia in development and cancer. And over the last years, we developed multiscale and multidimensional imaging techniques to study the role of EMT and cilia in mammary gland development and tumor genetics. And I will discuss that with you today. And I have to say that this investigation involves different members of the FBI from Rennes and Nantes. I will present data coming out from this platform using wide field, confocal, and light sheet microscopes used there. And I'm grateful for the technical help and simple advices from this fantastic group of people that I met along the way. So I will talk about the mammary gland, which is an organ that is made of ducts and lobules that are embedded in a vast stromal compartment that can be seen here, or that can be visualized here in our staining of the mouse mammary epithelium in green. You can see a duct and lobules here at the tip of the duct in the mouse mammary gland. The mammary epithelium itself is a stratified epithelium. It is comprised of a luminal compartment within the inside of ducts and lobules, and of a basal compartment, here stained in blue, and of a basal compartment on the outside of ducts and lobules that is stained here in green. Mammary gland development is a process that occurs throughout life. The mammary gland starts its development during embryogenesis, mid-embryogenesis, but it remains relatively rudimentary until puberty, a stage at which, under the influence of hormones, the mammary gland will undergo a first important phase of development until adulthood. And the mammary gland can undergo a second important phase of development, notably during the reproduction cycle, under the influence of hormones, again, from pregnancy to lactation. Then the mammary gland can go back to a quasi-virgin state through a process called involution. All of these developmental stages are controlled by distinct cell types. Multipotent mammary stem cells promote embryonic mammary gland development, and unipotent stem or progenitor cells of both compartments mainly control postnatal development. However, these stem cells can switch back to a multipotent state under regenerative conditions. In a similar manner than for development of the mammary gland, we know that the development of the different subtypes of breast cancers that are represented here, their development and recurrence post-therapy rely also on a subset of cancer cells called tumor-initiating cells or cancer stem cells. And thus, understanding the basic biology of the mammary stem cells and cancer stem cells is of key interest in developmental and cancer biology. And this goal has driven our work over the recent years. Important papers published over the last decade revealed that normal and transformed mammary cells activate an epithelial plasticity programs known as epithelial mesenchymal transition to acquire stem cell properties to regulate early mammary gland development or the formation of poorly differentiated breast cancers, triple negative or clodino breast cancers. The finding was rather surprising at the time of the initial discovery, since EMT, the process during which an epithelial cell can progressively acquire a mesenchymal phenotype, it was mainly described to confer invasive properties to cells during embryogenesis and to also a subset of cells within carcinomas to promote metastases. And despite this important new finding that connected EMT to stemness in normal and transformed stem cells for tissue development and the formation of tumors, the underlying mechanisms remain poorly studied over the last decade. And our research revealed relatively recently that EMT induced stemness in normal and transformed cells in the mammary gland and breast cancers by inducing a cell biological process called primary ciliogenesis, which is the dynamic process of assembling the primary cilium, which is at the center of our research. And we developed distinct imaging studies to really characterize the biology of this structure over the recent years. So the primary cilium is a microtubule-based structure that you can see here that is assembled at the cell surface in distinct cell types, distinct tissues. It depends on the tissue and the developmental stage. It is assembled by a modified version of the mother centriole of the centrosome that we call the basal body, and that can be seen here in this EM picture. At this location, the primary cilium is really a cell signaling center. It is well known that the primary cilium can control distinct signaling pathways, and key components of these pathways can localize to the primary cilium and are modified there, and therefore the primary cilium dictate cell fate during development and in adult tissues. And thus, it is not surprising to mention that dysfunctional cilia cause severe developmental defects as well as cancers that are represented here and that are called ciliopathies for the developmental defect. To go back to our tissue of interest, the mammary gland and breast cancers, we first showed that activation of EMT programs is associated with primary ciliogenesis in a subset of basal cells in the mammary epithelium, either in the mouse mammary epithelium or in the human mammary epithelium. As you can see here in our staining of this cross-section of mouse mammary gland, slug iris YFP mouse mammary gland, and of a human mammary gland. You can see cilia here stained in green through the staining of small GTPAs that localize within the cilium. Here, cilia protruding from centrosomes. We've showed after that activation of EMT programs is also associated with primary ciliogenesis in cancer stem cell-enriched tumor cells of Claudine-Low breast cancers, either in mouse tumors or in human tumors. And I have to say that we assembled a unique cohort of Claudine-Low breast cancer for that. And we developed strategies to stain primary cilia on sections of paraffin sections of these tumors that you can see here. So having defined that EMT is associated with primary ciliogenesis was nice, but at the time, the interesting question was how do does EMT programs promote primary ciliogenesis? And to address this question, we then used human mammary epithelial cells isolated from mammoplasty and immortalized in vitro, the HMLE cells. And we modified these cells to induce EMT and track ciliogenesis dynamically in these cells. And by using this model, we then showed that yes, indeed, EMT directly induced primary ciliogenesis, and that primary ciliogenesis is induced at intermediate EMT transition states. Having defined that, it was nice, but then the question was, how do EMT programs promote ciliogenesis, and what is the function of the cilium upon its assembly? Does it control stemness of normal and transformed cells? So to make a long story short, we developed distinct gene expression studies in models of mammary stem cells and cancer stem cells, and we developed 3D culture systems to address our questions, including a mouse mammary organoid model that derives from mouse mammary stem cells. And we also developed imaging techniques to analyze these organoids in 3D using light sheet microscopy. And we first showed that these organoids are composed of cells that express EMT-TFs on the outer layer of these rudimentary branches that you can see here that are ciliated, which makes it a good model to study our questions. And by using this system, we showed that EMT induced primary ciliogenesis by inducing FGF signaling. As you can see here, for example, inhibition of FGF signaling results in inhibition of ciliogenesis in these cells that are on the outside of the organoids. After that, we showed that once assembled primary cilia also regulate an important GLIAS2 signaling axis to promote stemness of normal and transformed cells. So, for example, here, genetic ablation of cilia or ectopic activation of GLIAS2 signaling results in inhibition of organoid forming capacity of mammary stem cells ex vivo. And we also showed that ectopic activation of GLIAS2 signaling in normal and transformed cells results in inhibition of their sphere forming capacity, which is another way to gauge stemness of normal and transformed cells. And we note here that the cells from these spheres that are non-differentiated cells are highly ciliated cells, as you can see here, stained in green, again, through an image through light sheet microscopy. So having defined that EMT-induced primary cilia and that the primary cilium regulated GLIAS2 signaling axis was super interesting to promote stemness. But the key question was now, how does the primary cilium-dependent GLIAS2 signaling axis control development and tumor genetics within the mammary gland? Briefly, the answer is yes. The primary cilium-dependent GLIAS2 signaling axis control early mammary gland development and the formation of clodino breast cancers. For example, here you can see that a genetic inactivation of GLIAS2 in the mouse results in severe developmental defects that can be seen at puberty here, that is associated with abnormal expansion of non-differentiated cells. We saw that by microscopy and also through gene expression studies. And for example, here ectopic activation of GLIAS2 and transform cells results in inactivation of their tumorigenic capacity. So this part is published, and so it revealed a novel aspect of EMT and ciliogenetics, but more generally, a novel aspect of the cell biology of early mammary gland development and the formation of clodino breast cancers. Now, in the time that I've left, I want to discuss with you unpublished results. The role of this EMT ciliogenetics axis and at later stages of mammary gland development remains as of now unknown, and also at later stages of breast cancer progression and during therapeutic resistance is also unknown. And we are studying this in the lab. So with regard to the first part, to investigate the role of EMT and primary cilia during the reproduction cycle, we've recently developed a biobank of mouse mammary tissue and techniques to analyze its development at distinct stages of the reproduction cycle. We collected tissue at the time when the tissue expands from pregnancy to lactation and at distinct phases of involution to find that the tissue starts only to regress 3 days after lactation, as you can see here. And then we started to develop techniques to analyze EMT and cilia in 3D in this tissue through clarification of the tissue and confocal imaging. We found that basal cells of the mammary epithelium undergo an EM to more M transition and then switch back to an EM phenotype during the reproduction cycle. You can see here the basal cells stained in red through keratin 14 staining. The basal cells start to elongate during pregnancy, but acquire really like a fibroblastic phenotype during lactation and switch back to a more EM state. We are looking here at basal cells that are surrounding an alveoli here in 3D, the place where the milk is produced within this alveoli. And these morphological changes are associated with the loss of an epithelial marker, Claudin 7, progressively from pregnancy to lactation in the basal cells, whereas luminal cells retain this epithelial and cell junction marker, and then it comes back during involution. So really suggesting that basal cells undergo EM to M and M to EM transition during this cycle. And then we analyzed cilia in this context as well. And strikingly, we observed that primary ciliogenetics is very dynamically regulated also during the reproduction cycle and in these basal cells specifically. We saw a pronounced induction of ciliogenetics exclusively in the basal cells during pregnancy. Then cilia are disassembled during lactation in the basal cells to be de novo induced in basal cells at involution, telling that primary ciliogenetics is induced more at an intermediate EM state during the reproduction cycle, specifically in basal cells. We saw cilia in basal cells from ducts and also from alveoli that you can see here, cilia stained in green in basal cells. We were particularly surprised to see cilia assembled here at involution a stage during which the tissue regress, as discussed previously. It's a time when a cell death occurs within the tissue, as you can see here through staining of CLIF-Cas3. However, we observed through FACS quantification that cell death occurs almost exclusively in luminal cells during evolution, but not in the basal cells that are ciliated. Then we hypothesized that cilia may promote survival of basal cells during mammary gland involution. I do not have the definite answer for that, but our first results strongly suggest that it is the case. At least the primary cilium promotes survival of mammary epithelial cells in vitro. Genetic ablation of cilia results in these cells results in induction of their death triggered by TNF-alpha, as measured here by FACS, or simply measured by video microscopy using a biosensor of Cas-7 activity, as you can see here. So to conclude on this part, we think that during the reproduction cycle, an hybrid EMT induced ciliogenesis in basal cells, and we are now developing strategies to investigate the role of cilia in stemness and life death decision of basal cells and in mammary gland remodeling. I have a few slides left. So regarding the last part with the role of cilia in breast cancer therapeutic resistance now, to address this, we recently developed a biobank of breast cancer tissue from 16 patients that were the tissue was collected at the time of diagnosis or after chemotherapy and surgery. We generated paraffin sections of this tissue to locate the tumors within the samples. And we developed also techniques to analyze EMT and cilia through immunofluorescence staining of sections. 3 minutes, okay. And,
including questions.
And so to make a long story short, we found that cilia are highly assembled in cells that are in an EM state, hybrid EMT, but we didn't find induction of ciliogenesis post-chemotherapy, 1 month post-chemotherapy. So we reasoned that ciliogenesis may be induced more dynamically during chemotherapy. And we developed organoids, so I will go fast, to analyze that breast cancer organoids derived from clodino breast cancers. We analyzed cilia in these structures. Again, through light sheet microscopy, we can see nice cilia within these structures here protruding from centrosomes. We found that these structures are comprised of cells that are in EM states, co-expressing E and M markers that assemble cilia that you can see here. And we found that cancer organoids that derive from chemotherapy-resistant cells are more ciliated. So we think that, again, an hybrid EMT may be associated, may induce ciliogenesis in cancer cells. And we are developing strategies now to analyze the impact on stemness and life death decision of these cells, and also on the role of cilia in breast cancer therapeutic resistance. And I thank you all for your attention. I thank all the people that contributed to this work that I initiated at MIT, I continued in Rennes, and now that I continue in Nantes. So
thank you very much. Yeah, we have time for one question. Yes. Effect of the cilia on the tissue, or is this just an antenna for
So we think that the mechanics of the tissue may influence ciliogenesis as well, mechanical properties of the tissue. With regard to the role of the cilia in inducing distinct mechanical properties, I do not know. We didn't study that. We think about the cilium as a real signaling center for transducing signaling.
Thank you very much. We need to move on. Yeah. So the last talk is from Alexandre Martinière from IPSIM, the Montpellier node. It's going to tell us about plant membrane nano-organization and osmotic signaling.
Right. Okay. Hello, everyone. So first, thank you for the invitation. I'm very happy to be here. So you will see I'm plant biologist, so it's a bit like unusual, I guess, for you. And I'm researcher, and probably one thing that I would to push here is how microscope and all those platforms actually help my research. So I will try to show you that. So I'm from Montpellier, and you probably noticed that plants they are quite fascinating in general. But one thing that really that I found is really interesting is that they have a continuous growth and development, which is obvious from here. And I think this ability is really interesting when actually it comes to extract nutrients and water from a complex resource like the soil is. So soil is really a complex structures with air bubbles and then salt particle that will constrain the localization of minerals and water. And plant, they use this continuous growth to optimize the uptake. And this is really true for water. So when you have a root system that grow in the soil, then it will go through many types of mechanism to optimize the uptake of water. These go from, I mean, the bottom right, from the tropism response to the creation of new organs to uptake water, and also to change deeply the anatomy of the roots. Something that is really striking is, whereas we know a lot about how water change the development of the root system, we don't really know yet how the plant cells are able to perceive if water is there or not. So a small recall about just a schematic or the main feature of a plant cell. So you probably remember it's actually surrounded by the cell wall, a complex mesh of carbohydrates. They have a very huge vacuole, so that is 90% of the volume of the cell, and this is a type of cell that is under Turger, which confer ability of growth. So the cell actually grow or expand because of the Turger. So obviously, if you change the water potential outside of the cell, you will have deep impact on the Turger of the cell, but also on the volume of the cell. So what we try to work on or research topic is really to understand better how the cell or the plant cell is perceiving water and how it gets integrated in growth and development. So what we did before is we found that when you stimulate roots with an osmotic stimulation to mimic a water stress, you have an increase of the reactive oxygen species that increase in the cell. And we use this as a readout to identify the molecular actors that are creating this accumulation of ROS. So we found that NADPH oxidases, that are plasma membrane localized protein, are really important for that. You may know that they are using the NADPH to transfer electron across the membrane, and then they get associated to oxygen and create superoxide. What I want to show you today is how those enzymes get regulated. And what we found is to be activated upon an osmotic stress, you need small GTPases. So in plants, we don't have the RAS. GTPase are not existing. We only have the raw GTPase. They are 11 isoform in Arabidopsis, and they have a big G domain as usual. They have a small tail that is contain polybasic amino acids, and they are prenylated at the C-terminus, and they have two sites of acylation. And as you know, there are molecular switches. They exist in two states, one active and inactive form, and they obviously have the GEF and the GAP protein in plant 2 that actually control the two states of the GTPase. So first thing that we did is that we screened for all the raw GTPase that are expressed in the roots, and then we used this cross accumulation to see if they were implied in the signaling that we are studying. So here are just small results to show that if you apply an osmotic stress, you're able to record an increase of fluorescent signal that is coming from a dye that increase the fluorescence when it gets oxidized. And what is interesting to notice is if you look at a plant where the ROP6 isoform is not expressed, the protein is not there, then we don't have this accumulation of ROS. We generated overexpression line, and then we found that this is the quantity of the protein is not really limiting. So what we believe it's the activation of the GTPase that is important, and this is shown on the graph here, where we use point-mutated GTPase that are locked in the active form, and then we complement the mutant. And probably here you can appreciate that in this situation, the plant is having a constitutive accumulation of the ROS. Last thing that we did is that we check where the gene is actually expressed. That's what you can see. I'm sorry, I don't have pointers. So it's on the left side. So you can have, I mean, at the tip of the root system, then the gene is there. And also what I try to highlight is that we also have it expressed where the lateral root will be formed and then later emerge. So it's exactly where development of the root system is changing in response to water stress. Then, if you go back to textbook, what people said about GTPase is that they are usually activated at the membrane and they are sequestrated in the cytoplasm when they are inactivated. So then we were wondering if is it also the case for plant GTPase or GTPase. And then when we look at the localization of those proteins, either when they are locked in the GTP form or locked in the GTP or GDP form, or after signal stimulation, we didn't find any effects. So here, the blue pictures is basically showing the plasma membrane of the cell. And then if you look at the cortical part of the cells, you don't see neither any structures or any change of localization. So then we were wondering if maybe within the plasma membrane, the dynamic of the protein might be touched. So then we did FRAP experiments. And what we found during FRAP experiment is if you stimulate the cell with an osmotic signal, we were able to see a change in the curve of the recovery. So those are control experiments and cells after stimulation, and then we were able to see a change in the dynamics. So maybe something was actually happening. So then we get contact with experts, and then we had a try on the TUF microscopy. That have like a better signal over noise ratio and really get nice picture on the membrane. And then what we found is that if you increase the osmotic signal minutes after the treatment, it was really obvious that the organization of the signal totally changed, and we start seeing like small dots with medium signal, medium stresses. And then if we increase the concentration of the osmotic signal, we had more cluster forming. We also found that those structures, they were pretty static, as you can see on the chymograms, and they were very small because if you try to look at their size, they were more or less at the size of what is expected for the diffraction limits. So then we said, well, that would be interesting to go even further and then to have more information about the diffusion and also the localization of those proteins. So then we did SPT-PALM on the plants. So here there are a small movie, but I'm sure most of the audience, they are really used to those things. So that's what you look like in plants. So probably something similar to what you see in animal. Those are really single molecules. Obviously, we use them to do track reconstructions. Then we extract information about diffusion. And we were really happy to see that when we stimulate our cells, we had like a massive effect, quite striking effect on the diffusion. So that's what you can see on the left side, The green curves are the diffusion of the control situation where you have clearly two populations of molecules. And then if you stimulate it, you have an increase of the number of molecules that have low diffusion, right? We did also some tessellation to have some information about the clustering. Obviously, after that, I was really excited. And then we said, let's make a link between the activation of the protein and the behaviors. So we use again this log version of the GTPase, this point mutated version. And then we found that without any stimulation, the log version of the GTPase is show a high proportion of molecules with low diffusion. That's what you can see here. Then we tested also the role of this acylation. Remember, I told you that there are two sides of acylation in the protein. So it's quite known that acylated protein, they have specific partitioning with lipids. So then we were wondering if those acylation sites might be important for the dynamic of the protein. So we mutated the protein. We did PALM on it, and then we found that sorbitol, the osmotic treatment, had no effect on the dynamic of this mutant. And then if you complement the Arabidopsis mutant with this mutated version of the GTPase, we were not able to recapitulate the ROS induced by osmotic treatment, meaning that those sites are probably important for the signaling. Right. The last step for us was to try to confirm our system. As I tell you, we were suspected those GTPase to regulate the NADPH oxidase, so this enzyme that produces the superoxide. So we did two experiments to try to push this idea. The first one was to record the thread between the two fused proteins, so the RBOH, so this is the NADPH oxidase and our protein, and we record this thread by FLIM, and we found that we have a decrease of the lifetime after treatment with the sorbitol that was not observed in the control there. And then it was in the same frame of intensity that what we have with the version of the GTPase. The other information that we found is that the GTPase, the NADPH oxidase is also making cluster after stimulation. And this is probably not the best piece of data of ever, but we try to do a TUF2 color imaging. And then it seems that they might actually be associated in the same structures. So I will give you two other pieces of information that go a bit further than this work. So this is the model that we are working now. So we believe that this osmotic signal is changing the localization of the GTPase, even if it's not really demonstrated straight, even the activation is not demonstrated. We believe that activation is needed and probably acylation, and this controls the ROS accumulation during osmotic signal. But what was really interesting is that meanwhile we were doing that, colleagues in Lyon was working on another signal on the same GTPase, which is the auxin, which is a very important hormone for plants. And they found basically the same thing as us, is that it's clustering the GTPase, the same isoform in the same subtype as us. So then we were wondering if the protein that co-clusters, so which this NADPH was also present after an auxin treatment, and it was not the case. So if you do an auxin treatment, you see the dotted structure, you see dotted structure in sorbitol. And now if you take the NADPH today, and you treat it with sorbitol, and then if you treat it with auxin, you don't see any cluster. So what we believe now is depending of the upstream signals, then the clustering, the protein, the effector protein that co-cluster with the GTPase change. Last bit of information. So we also work a bit on the role of this polybasic tail of the GTPase and its role of interaction between anionic lipids. So what was found is this charge part of the protein is interacting with phosphatidylserine. And we also found that the quantity of this phosphatidylserine at the plasma membrane and its organization really steer the strength of the response of the GTPase. More PS you have, more strong response in terms of signaling you will have. And more recently, we discovered the mechanism that might control this quantity of phosphatidylserine at the membrane. And this is mediated by a receptor kinase that perceived the cell wall status, and then we'll transfer this information to phosphatidylserine localization and quantity. So what comes next for us? So the two main questions that we are trying to push is how the signal specificity is controlled. So we are working on that at the moment. And obviously, we would really love to take this knowledge and try to push it more in terms of the development. So what is the impact on the, at least on the cell growth? That's something that we try to do. We are currently developing new tools to understand better our systems. So two things. So the first thing is to use or to develop a rope activation sensor on the base of what is known in animal fields. That's one bit. And the other thing that we want to use is to use sensor for H2O2, which is a side product of superoxide production, to have an information about the localization of this host accumulation. So with that, I just want to thank all the amazing people that I had the chance to work with, especially Maria, that was doing a PhD with me, that did most of the work that I presented, our collaborators in France and in Germany, and obviously all the platforms. So especially I want to thank the group of Marcelo Nollmann and Jean-Bernard Fisch, where we did most of the SPT Palm was done there. Also, just for the small story, I actually start all this SPT Palm on plants in Bordeaux, in the group of Daniel Choquet. And then we moved and we set a collaboration in Montpellier because it was obviously easier to do the experiment. And with that, thank you for your attention. And if you have any question, I will be happy to answer it.
Yeah, for some reason, yeah, does work. Yeah, we have time for questions. Yeah, Pierre-François. Ah, pardon. Wait.
Thank you very much. Just a technical point, or you managed to make turf on plant, and you have the cell wall, how it works?
I don't know, but it works. No, well, it's interesting. I think, well, I did turf even when I was postdoc, even before coming back to France on plant samples. And indeed, I mean, all people that you will talk with will tell you this will never work because of the cell wall thickness. What I would guess is well, it's a question for you and not for me. It's working, we are sure about that, but maybe one thing that might happen is that the cell wall might have the same refractive index as the glass, I wonder, and maybe the evanescent wall will then form at the membrane like it is.
But you're sure you're in turf? You're not doing just high-low illumination?
So we try, well, it's from what I understand, it's quite complicated to really know if you're doing turf properly or not. Mm-hmm. What I can tell is that we clearly see refraction of the light of the illumination. This is really really clear. And then if you use commercial setup, and then you have like something to change, and then the machine tell you are using turf. So then the machine tell maybe it's true. We are using turf then.
So yes. So one remark about this. I mean, we did super resolution in tissue in embryos, and we are clearly in high-low illumination and not in proper turf. But the important is that you get super resolution at the end.
Yeah,
so.
Sure. This is and, really an important point.
My question is related to what, so your conclusion about the specificity of signals. I suppose that if you have an osmotic shock, it will change the dynamics of many molecules. So if you look at molecules that you know interact with the membrane, which is not a raw GTPase, another molecule, do you see signatures, similar signatures of changes, dynamics, association?
It really varies depending on the protein that you're studying. So I mean, these GTPase behave like this. If you're looking at aquaporins that are, poor for water transport, they have not the same signature, and we have also protein that are not changing their diffusion upon the same treatments. So we believe this is really something that is linked to a molecular signal, not a broad response of the membrane.
Thank you. Yeah. Do you have other questions? No. Maybe in general, could you tell us in your mind what's the major hurdle of doing single molecule imaging in plants?
Well, before starting, I would have said that the main difficulty is actually to get data out of the microscope, and this was really easy. So we just, because we are working on full organs, I mean, we just take the roots and then we look at the epidermal cells of the roots. And this was really easy to actually generate data. Then the main thing for us was really to understand deeply, or at least as much as we can, the analysis below and what we can extract exactly, how we are making it, when are we making errors, and, how to set properly the tracking, all those things was a bit of more work, I would say.
Okay. All right. Thank you very much. Thank you. And thank you to all the speakers. Congratulations on sticking exactly to your times. And
to the chairman. And we'll be back then at 1:30, please. And if you want to let stuff here during the lunch break, I can close the room. So.
Pardon?
Des choses que tu...